This is the second video on macromolecules. In this one I want to talk briefly about proteins and enzymes, which are a protein with a special job. Proteins are composed of the elements carbon, hydrogen, oxygen, and nitrogen, and there also are a few proteins that are going to have sulfur in them as well. The monomer, the building blocks for proteins, because proteins are a polymer, again, a very large molecule made up of a repeating unit, and that repeating unit is amino acids. Now there's actually 20 different amino acids that are linked together in different orders to make all the different proteins in the body, and you can see that with 20 different amino acids to play from, you can get a whole variety of proteins. Protein is very important for sustaining life. It is essential for cell structure, and because it provides the structure for cells, it ultimately provides the structure for the organism. Protein is necessary for movement, both inside the cell, microscopic movement. There are proteins that are responsible for moving things around, but also gross motor movement. The things we think about our muscles moving, well, it's protein fibers in the muscles that allow our muscles to contract. Proteins also make up some of the hormones. An example of this is adrenaline, also known as epinephrine. We know that some of the proteins are lipid-based, but there's another whole class of hormones that are protein-based. And then proteins are enzymes, which are proteins with a particular job to make a reaction happen, a chemical reaction necessary for life that wouldn't happen fast enough without an enzyme. An example of this would be amylase, which is the protein found in your saliva and also in your small intestine, which will break down starch into its individual glucose molecules. So let's talk a little bit about amino acids. All of our amino acids have a basic structure that is the same for all of them, and then a distinct side chain, which is known as the R group. So the basic structure consists of a central carbon atom, an amino group, and a carboxyl group. And so if we look at this diagram over here, we see our central carbon. On one side of it, we're just attached to a hydrogen, like we see over and over again in these macromolecules. And on the other side, that's where the R group attaches. On the one side, we have the amino group, which is a nitrogen with the hydrogen, two hydrogens attached to it. And then on the other side, carboxyl, or the COOH group. Different side groups in the amino acids give each amino acid its distinct characteristics. For example, we have two different amino acids shown down here. Leucine, its side group is hydrophobic. It doesn't like water, whereas serine, it is hydrophilic in its side group. So that's one thing that will distinguish one amino acid from the other, is what is the side group and what characteristic that side group has. Just like all of our other macromolecules, amino acids are linked together through dehydration synthesis. And so this process that takes a water molecule out to cause a bond to form, to put two monomers together, works for proteins just like we saw working for carbohydrates and for lipids. We call the particular bond a peptide bond. The word peptide itself just kind of refers to protein in general. Here it is a carbon bonded to a nitrogen, not a carbon to an oxygen like we saw in carbohydrates and lipids. So in our little diagram down here, we have one amino acid needing to bond to another amino acid. You can see we've just got an R group, so we're not quite sure what this amino acid is. But again, our OH group from one and a hydrogen off the amino group are what are going to come together to make the water. Over here on the right is the products. And then our carbon-nitrogen bond here, the peptide bond that holds those two amino acids together. And so a protein is made up of more than two amino acids. It's many, many amino acids, hundreds, thousands, some of them even tens of thousands of amino acids. And so they can be thought of as just sort of a long chain of amino acids all hooked together to make that very large molecule. But it doesn't just stay as a chain. So peptide chains, or polypeptide chains, will fold and bend into a particular protein's functional shape. Remember, form follows function, and so these proteins have a particular shape. And basically there's 4 different stages of how a protein can bend. We call the primary structure this amino acid chain that forms, and that's what's going to happen at the ribosome when the protein is being put together. But really, as soon as you have a chain of sufficient size, it's going to start to twist and bend as hydrogen bonds form between the hydrogen and the oxygen, or the hydrogen and the nitrogen present in those amino groups or in those carboxyl groups. And so when it forms into this sort of swirling, spiraling shape, it's known as an alpha helix, or if it can fold in sort of a bended shape, a more flat kind of structure, it's a pleated sheet. And so both of these are known as secondary structures. They're happening after the chain has been made, and you can see we've got hydrogen bonds forming here between nitrogen and hydrogen, and on this other one between the oxygen and hydrogen. Hydrogen bonds are not as tight as a covalent chemical bond, but they're strong enough to hold molecules together. It's what holds those 2 strands of DNA together. And then the R groups come into play. And R groups that are alike tend to kind of attract each other. And so R groups that are hydrophobic or water-fearing will collect together, and they typically do kind of form in the middle of the protein. Proteins kind of end up as this sort of globular shape. And so the different R groups will cause it to bend and twist around into what's known as the tertiary structure. And then some proteins are more than just one amino acid chain, and so there's 2 or more. Insulin, for example, are 2 polypeptide chains bent around each other in just the right way. Hemoglobin in our blood are 4 polypeptide chains, 2 of one kind and 2 of another, all twisted together. And so when you have more than one polypeptide chain going into that protein, then you end up actually with a quaternary structure, sort of the 4th level structure that gives that protein the exact shape it needs to do what its particular function is. Now, the substitution of even just one amino acid can cause a disaster, or close to a disaster here. In the sickle cell disease, that is caused by one wrong amino acid in that original protein polypeptide chain. And that causes, instead of a normal red blood cell that's sort of like a donut, you get a sickled or a crescent-shaped red blood cell. And you can see that the problem is our amino acid number 6 here, instead of glucine, we have valine substituted in. And just that one amino acid with its one R group that doesn't really do what the glucine did originally, it's got a different characteristic, causes the protein to have a slightly different shape, which causes the red blood cell to have a slightly different shape, and which causes a disorder and pain and suffering for the person who has sickle cell disease. Of these amino acids, 9 of them are known as essential, and they must be obtained from food. So you do have to get them. The other amino acids can be manufactured in our body. They actually are subdivided into non-essential amino acids, ones we can always make, and conditionally non-essential, which sometimes if we're under a great deal of stress or we're suffering from a particular illness, we may need to get these from food. We're not able to manufacture them. But the essential amino acids have to come from food, and animal products do tend to give a better balance of these, so that's why it's just easier to get your essential amino acids from animal foods. It takes a little more planning to get them from plant foods. You want to have a regular protein intake throughout the day. The standard American diet tends to heavy load dinner time as a place where we get lots and lots of protein, but not so much at breakfast. And it's actually better for our bodies if protein is relatively evenly distributed across the day. And one of the things that happens with aging is that protein needs actually increase as someone gets older because the body is less able to efficiently use the protein that they actually take in. So your grandparents actually may need more protein than you need, even though you think you're young and energetic and have lots of using your body a lot and muscles a lot, but actually your grandparents are not able to absorb the proteins they eat as well, so they need to take in more to begin with. Now I want to turn to enzymes, which are a particular kind of protein that acts as a biological catalyst. And a catalyst is something that speeds up a reaction without being part of that reaction. Enzymes then increase the rate of chemical reactions that are needed for life. A lot of the reactions that are going on inside organisms' bodies, inside our bodies, if left on their own, might happen, but not fast enough as what we need in order to stay alive. So we need these enzymes. They're very, very important. Enzymes are also specific for only one chemical reaction. So you can't, you don't have an all-purpose enzyme. You have to have just the right enzyme for this reaction and just the right enzyme for that reaction and another enzyme for another reaction. And so because of that, thousands of enzymes exist. Each enzyme then matches its particular substrate or the reactants that you need for that chemical reaction that it is to catalyze. The substrate fits into what's known as the active site on the enzyme. So remember, enzymes are proteins. Proteins are long chains that fold around and sort of make this lumpy shape. And so there's a place where there's an attraction between the atoms in the molecules of the reactants and the atoms of the protein that pull those two pieces together. Once the reaction, or once the substrate is inside the active site, the reaction can occur, and then the reactants become products, just like any other chemical reaction. These products then will detach from the enzyme, and the enzyme is free to match to new substrates, so it is not changed by the reaction. It is used over and over and over again. So enzymes do this reaction at different speeds. The fastest one I have read is one that occurs about 100 times a second, but most enzymes work a little bit more slowly than that. So here's an illustration of that whole process. So you have a substrate coming into the enzyme, matching up. So you see how we have these shapes here that are going to match the shape of the substrates. And then as they come together, the enzyme is believed now to actually sort of change shape slightly. We call that an induced fit. So that's instead, it used to be called the lock and key, thinking that it was already perfectly formed, and now the theory is that it's an induced fit, that as the substrates come in, the enzyme sort of shifts a little bit, and so till it fits perfectly. And then the enzyme will do its thing, whether it's these two, in this case, we have an example of decomposition happening where we have a larger molecule going to be turned into two smaller molecules. And so the enzyme is bringing just the right parts together to allow this to happen. And then the products leave, and the enzyme is then available to react with another substrate if another substrate is present. Enzymes can be divided into two broad categories. We have anabolic enzymes, which are ones that build more complex molecules from the substrate. And so dehydration synthesis, we've been talking about it, puts smaller monomers together to make that larger polymer. But also DNA replication, when we build a new strand matching an old strand, when cells are dividing and they need to make a new copy of the DNA, well, there are a whole bunch of anabolic enzymes that are involved in building those new DNA strands. And then we have catabolic enzymes, which do the opposite. They break those larger molecules down into smaller molecules. Of course, digestion, where we see the process of hydrolysis being a prime example of that. But also, your body is doing a lot of recycling. There are dead cells that need to get taken care of, or molecules that are not functioning anymore, and they need to be broken apart, and enzymes are needed for that as well. And finally, enzyme function is greatly affected by temperature and pH, which is those two situations of homeostasis where your body works very hard to keep your body within a limited temperature range and very, very tight control on pH of body fluids. Because if these get out of balance, then the enzymes that are there keeping you alive by helping all these chemical reactions to happen are no longer able to do their function, and then you are no longer able to stay alive in the worst case scenario. This finishes the overview of proteins and the particular type of protein known as enzymes. There's one more video in this macromolecule series talking about nucleic acids.